not last week, this week only we have discussed about the external features of brainstem. Okay, if you remember, I have written the points under which we are going to discuss the brainstem. Okay, I don't know whether you have noted those points or not, but those points were very important. You have to understand how to study the brainstem. Okay, in the last class, as I mentioned, we have studied the external features of brainstem mainly, where we have seen the introduction, then important function as a whole, okay, then subdivisions of the brainstem, followed by the external features. Can anyone tell me how to study the external features of brainstem? How you will study the external features of brainstem? Two views are there, anterior view and posterior view. Okay. Now suppose you are taking brainstem in your hand, then how you will study? First of all, the anterior view. सामने से क्या दिखाई देता है, ठीक है? तो what are the external features, and then from the posterior view. This is the best way and easiest way to study the external features of brainstem. Next, that day I have mentioned last 10 cranial nerves are attached to the various part of the brainstem. Okay, and along with the diagram I have shown you from, and each and every nerve from which particular part of the brainstem they are arising, that we have seen, that we have studied. Okay, now we are going inside the brainstem. Okay, we are going inside the brainstem. So in order to study the internal structure of any organ, any structure, what do you do? If you want to study the internal structure of any organ, what you used to do? Internal structure के लिए क्या करते हो? What you have to do? You have to cut it. Okay, so we can't cut like butcher. We have to be very specific. So different sections are there. With the help of those sections, we have to cut the structure so that the internal structures which are present, okay, that will not get damaged. Okay, if a object is there, we are cutting it in such a way that the internal structures are preserved, then only we can study the internal structure, or else we can't study. Okay, so for that we can't cut it anyway, whatever we want. Particular sections are there. So in case of internal structure of brainstem, how to study the internal structure of brainstem? Okay, so in order to study the internal structure of brainstem, what we have to do? First of all, we have to take TSC. This topic is very important, plus it is very difficult topic. Okay, so I'll request everyone to please listen it carefully. In the upcoming exam, you can expect some questions from this topic. Okay, so here first of all you can see you have to take TS at various levels. Now that is specific. You can't take TS at the level whatever you want. some specific points are there. In that particular point only you have to take the TS. Okay, and it varies from part to part of the brainstem. For example, in case of medulla, the parts are already mentioned, the TS are already mentioned, so we have to take the TS in that particular part only. Okay. Now, once you take the TS at various level, okay, what you will see, the interior can be seen. Now, the interior is divisible into two parts. Okay, that is gray matter and white matter. Now, in case of brainstem, you will be getting both the part. You will be getting the gray matter also, you will be getting the white matter also. Now, in gray matter mainly you will see different nuclei are present. Okay, so you have to mention the name of the nuclei. You have to know the name of the nuclei. Along with that, you have to know the position of the nucleus. Now that means whenever you are taking a TS, okay, you have to know which part is the anterior part and which part is the posterior part. Now, do you remember the TS of spinal cord? TS of spinal cord already we have discussed. Now, how you will come to know that which part is anterior, which part is posterior in the TS of spinal cord? Yeah, based on the fissure and sulcus. Fissure is present anteriorly, sulcus is present posteriorly. So, this is an important identifying landmark. Similarly, in case of brainstem also, first of all, when we are taking the TS, we have to identify which part is anterior, which part is posterior. Okay, if we don't identify that, we will not be able to understand the relations of the gray matter and white matter, which is very important. This is the first thing. Next, what we have to do, always keep that in mind, you have to segregate the structures. Two types of structures are there, gray matter and white matter. Gray matter में क्या रहेगा? What you will see in the gray matter? Different nuclei are present. Okay, size may be different, but mainly nuclei are present. So you have to know where exactly the nuclei are situated. Okay, what is the position? Whether it is ventrally, whether it is dorsally, whether it is in the middle. Okay, so you have to know the position. Next, about the white matter. In white matter, two things are there. Okay, first of all, you have to know about the fibers. That means ये जो nuclei है. See, I have mentioned about the nuclei. So nuclei means the fibers will come out from the nuclei, right? So you have to know which fibers are coming out from which nuclei. Hello, go to library. Go to library. So in case of white matter, as I said, first what you have to know, first you have to know about the fibers. ये जो particular nuclei है, from this nuclei the fibers which are coming out, that you have to know. And along with that, the tracts, you have studied about the tracts. So the tracts which you have studied in spinal cord, okay, they are present in the brainstem also. So what is the location of the tracts in the brainstem, that you have to know. Okay, so these are the two important points which you have to mention in the white matter. Okay, next, here I have mentioned important connections. Now what is the meaning of this? When you will take the TS, at that time you will see so many nuclei are there. Okay, multiple nuclei are there. ऐसा नहीं है कि सिर्फ एक ही nuclei है, multiple nuclei are there. Each and every nuclei, they are having fibers. Okay, so among them, what will, and it's not possible to explain everything. So what I will do, I will select some important nuclei and we'll discuss the connections. Okay, now always keep that in mind. Whenever you are mentioning the connections, you have to mention the afferent fibers and efferent fibers. Okay, in important connections means what? Important connections means the connections of this particular nuclei. Okay, so in that you have to mention afferent fibers and efferent fibers. तो मान लो एक particular nucleus है, तो कहाँ से fibers आ रहा है? From where the fibers are coming, and then the fibers which are coming out from the nucleus, where they are going. So the fibers which are coming, incoming fibers, these are called as afferent. The outgoing fibers, these are called as efferent fibers. Okay, if you are not mentioning these two, your connection is incomplete. You can say that it is just like a circuit. जो electronic circuit होता है ना, same like the circuit. Both the components should be mentioned. Okay. So we'll start with the internal structure of medulla. Okay. Now there are some prerequisites which you must know before going to the internal structure of medulla. What are these prerequisites? First of all, if you see the internal structure of medulla, lower part of the medulla, it is almost similar to that of the spinal cord. So already you have seen the internal structure of spinal cord, right? The horns which are there, central gray matter, central canal, then peripheral white matter. So if you see the structure of spinal cord and the lower part of the medulla, internal structure, it is almost same, but there are some differences. Now what exactly those differences are? First of all, the internal structure of the medulla, white matter is surrounding the central mass of gray matter, but the gray matter is broken up into nuclei. Here, instead of getting the horns, you will see that the gray matter is broken up to form different nuclei. Okay, very specific nuclei are present, because we have seen that the cranial nerves are coming out. So specific nuclei should be there. Okay. Now the gray matter is broken up into nuclei that are separated by the nerve fibers. So the nerve fibers which are there, what they will do, they will separate the nuclei. Okay. So this you can expect in the internal structure of the medulla oblongata. Next, now there are different levels. So do you remember in the first class I had mentioned total 8 sections are there. Total 8 sections are there, okay, of brainstem. How many are there in the medulla? 4 sections are there in the medulla. How many sections are there in the pons? 2 sections. In midbrain, 2 sections. So total kitna hua? 8. Okay, so 8 sections are there. Okay, so we'll see one by one. What I will do, I will not explain all the sections. Okay, important sections will be discussed. Next, remaining sections study from your own. Okay, practice the diagram and study from your own. In that, if you are having any doubt, you can come and ask. Okay. So here you can see first section is at the level of pyramidal decussation. I hope you remember the pyramidal decussation. The pyramid where the pyramidal fibers are decussating, means going opposite to each other. Okay, at that level you have to take one section. Okay, though the section is passing through the pyramidal decussation, okay, that's why it is also called as decussation of pyramid. So at that level we have to take one section. Second section we have to take at the level of medial lemnisci. Anyone, what is this medial lemnisci? Don't use gadgets unnecessarily inside the classroom. Please maintain the decorum. What is the meaning of medial lemniscus or medial lemnisci? Anyone? In physiology, have you studied about this? Can you tell me what are the different types of lemnisci present? Okay, we'll discuss what are the different types of lemnisci present. Okay, now please keep that in mind. Now this is the decussation of pyramid, that means the decussation of the pyramidal fibers. Now here the decussation of medial lemnisci means the decussation of sensory fibers. Okay, the sensory fibers which are coming from the body, they also decussate in the midline. Okay, and after that they are forming the medial lemnisci which ascend. So that is the medial lemniscus. So at this level the section has to be taken. Next, another section where another level where the section is taken, that is at the level of olive. So in case of external features of medulla oblongata, you have seen pyramid is there. If you go posterolateral to pyramid, olives are there. Okay, so at that level also we have to take one section. Okay, and lastly at the level of pontomedullary junction, just the junction between the pons and the medulla. So these are the various levels where the sections are taken in order to see or in order to study the internal structure of the medulla oblongata. Okay, now we'll start. So first let me show you the diagram here. So this is the diagram. Have you seen this diagram earlier? Anyone? Okay, no problem. Now this is the TS at the level of deposition of pyramid. Okay, so this is how the diagram will appear. Okay, now how you have to study, if you see so many things are there in one particular diagram, how to simplify it so that you can understand. कैसे simplify करना है? See, I have mentioned for that. First you will take the TS at various level. So at which level you have taken the TS? At the level of pyramidal deposition or the deposition of pyramid. Next what you have to do? You have to identify the gray matter. To identify the gray matter over here. Can you tell me which is the gray matter? So this entire part is the gray matter. Okay, and this entire part is the white matter. So first you have to segregate the gray matter and white matter. Okay, that's why whenever you are preparing notes, please use two different colors. I am not telling in the time of exam. When you are preparing notes, okay, please use two different colors to show the gray matter and white matter. Okay. So here what you have to do, this is the gray matter and this is the white matter. So you have to segregate. After doing the segregation, what you have to know? You have to, in gray matter, what you have to write? About the nuclei. Okay. So you can see so many nuclei are there. So you have to mention about the nuclei. Okay. Next, once you have mentioned about the nuclei, then as I said, there are some important connections. So you have to mention those important connections, and that is the most difficult part. Okay. Now diagram you can by heart, or if you practice twice thrice you can draw the diagram. The most difficult part is whenever you are mentioning about the nuclei and their connections, because each and every nuclei, they are having different connections. Connections are not same. Now again there is a shortcut. Okay, in order to save your time during the time of exam, क्या करना है? Suppose you are drawing the diagram, so what you have to do, what you can do, you can draw the diagram of one half only. Okay, you can draw the diagram of one half only. But if you are drawing the diagram of one half, you have to show all the points, means all the labelings must be shown. Okay. Now one difficulty arises is, suppose you are drawing one half like this half you have drawn or this half you have drawn, then what will happen? The deposition cannot be shown. Okay, you can't show the deposition. So what I will request, the section where deposition are absent, जहाँ पे deposition नहीं रहता है, in that case you can draw one half only. Where the depositions are present, please draw both the half. Okay, so we'll start with the gray matter. The first gray matter. Okay, before going to the gray matter, which is the anterior side and which is the posterior side over here? Very important. Yes. So this is the anterior side and this is the posterior side. ये क्या है? What is this? Fissure. Very good. And what is this? ठीक है? So that concept must be clear. Now this you are able to answer because you have studied the spinal cord. Once we go into the Pons, things will get complicated. Okay, because in Pons you will not get the fissure and you will not get the sulcus. At that time the features are different, the identifying features are different. Okay, here though you know, I have not, that's why I have not mentioned again. Okay, so we'll go one by one. So first we'll come from the dorsal aspect to the anterior aspect, especially in case of the gray matter. Okay. So first of all, you can see the nucleus gracilis and nucleus cuneatus appear as a narrow strip-like projection from the posterior aspect of the gray matter. From the posterior aspect of the gray matter, you can see narrow strip-like projections are there. Okay, nucleus cuneatus and nucleus gracilis, which can be seen very easily. Okay. Next, the apex of the posterior horn. Now, this is the posterior horn. So apex of the posterior horn gets swollen to form the spinal nucleus of the trigeminal nerve. Very simple. Okay. First of all, strip-like projections of central gray matter on the posterior aspect, that is called as nucleus gracilis and nucleus cuneatus. Okay. Next, this is the posterior horn. This is anterior horn, this is the posterior horn. The posterior horn is swollen to form the spinal nucleus of the trigeminal nerve, which you can see in the diagram. Okay. So here diagram is important. If you know the diagram, you can write everything. Next, so this spinal nucleus which is formed over here, that is nothing but the continuation of the substantia gelatinosa. Okay. Next, the spinal tract of the trigeminal nerve is also seen. Here you can see. Okay. तो क्या होता है, जो spinal nucleus होता है, just above the spinal nucleus, the fibers are present and which is in the form of cap covering the spinal nucleus, and that is called the spinal tract of the trigeminal nerve. Okay. So these are the structures seen over here, specially, okay, in the gray matter. Okay. So gray matter at this level is very simple. Only three nuclei are seen. What are these? Nucleus gracilis, nucleus cuneatus, and then spinal nucleus of the trigeminal nerve. And here you can see this is the spinal tract of the trigeminal nerve. Okay. Next, other nuclei are also seen, but here the way to study those nuclei are different. Okay, and I am coming to that. Next, we will be focusing on the most important striking feature of this particular section, that is the decussation of the pyramid. The most important feature. So approximately 75% of the fibers, okay, are decussating. So where they will go? They will run backwards medially. As you can see, they are running backwards and medially. Okay, and at the midline, the decussation is taking place. Okay, you can see in the midline the decussation is taking place. Okay, of the opposite side. Now, in doing so, when the decussation is taking place, at that time, what happens? The fibers, they detaches the anterior horn. ये देखो, the fibers are coming like this, and the decussation is taking place. Now, these particular fibers, when the decussation is taking place, at that time, what they will do? This anterior horn is detached. From the central gray matter. Okay. So this detached anterior horn of the central gray matter, what they will do? They will form two different nuclei. Can you tell me which two nuclei are formed? Which nuclei are formed? That is the spinal nucleus of the accessory nerve. Okay, spinal nucleus of the accessory nerve and supraspinal nucleus of the first cervical nerve. ये जो part detach हो रहा है, this detached part, okay, of the gray matter, central gray matter, especially the anterior horn, will give rise to two nuclei. First is the spinal nucleus of the accessory nerve, that is the 11th cranial nerve, okay, and a supraspinal nucleus of the first cervical nerve. Okay. Now this, when it is detached, the supraspinal nucleus which is formed, okay, the supraspinal nucleus will be continuing as a hypoglossal nucleus, okay, and the spinal accessory one, the nucleus of the spinal accessory, that is extending up to 5th cervical vertebra. Okay, that will be extending up to the 5th cervical vertebra. Next, here you can see the appearance of the reticular formation also, okay, which is nothing but a diffuse form of fibers and nerve cells are present, that is called as reticular formation, which controls the consciousness of a person. Okay. Next, the other features, especially the tracts, okay, in this region, these are same as the spinal cord. Okay. So see, like this we have segregated. First, what we have discussed, about the gray matter. In gray matter, what we have seen? We have seen the nuclei. Okay, what are the different nuclei? Next, what we have discussed? We have discussed about the white matter. In white matter, the most important feature, that is the decussation of the pyramid. Once the decussation is taking place, we have seen that the anterior horn is detached from the central gray matter and two other nuclei are formed. Okay, so up to that much. Next, here you can see these are the tracts. On both the side. So the location, or you can say the position of the tracts over here is same like the spinal cord. Okay, so you can use that particular line. Means particularly you don't have to mention that which tract is present in which area. Just you have to mention, you can mention that the tracts over here are same like the spinal cord, the position of the tracts. Okay. So that is the pyramidal decussation. Next section is at the level of sensory decussation. Okay. Now here also with the help of the diagram we will discuss. Here you can see TS at the level of sensory decussation. Can you see the decussation of the fibers? See this one, these fibers. Okay. So this is the sensory decussation. Or now in sensory decussation, so what you can see over here, so pattern will be same. Okay, in sensory decussation also pattern will be same. That means first we have to identify the nuclei. Okay, now here can you tell me which is the anterior aspect, which is the posterior aspect? Here which is the anterior aspect and which is the posterior aspect? So this is the anterior aspect, this is the posterior aspect. Okay, now why I am calling this as anterior aspect? See, because here you are getting the pyramid. Okay, you are getting the pyramid. So in Medulla Oblongata, where the pyramid were situated when we have discussed about the pyramid. Now here what we have to do, as I said, you have to know which one is anterior and which one is posterior. Okay, so here you can see these are the pyramid. That means this is the anterior aspect and this will be the posterior aspect. Okay, because if you have studied the external features of Medulla Oblongata, in that you have seen anteriorly the pyramids are present. Okay, so that means this is the anterior aspect, this is the posterior aspect. So every time whenever you are studying about any decussation, any TS, first must know that which is anterior side and which is the posterior side, or else what will happen? See, so many structures are present over here, you will not understand the relation. Okay, and here we will be using some relations, that's why I am mentioning. Okay, so first what you have to do, we have to study the grey matter. So in grey matter what you can see over here, especially if you go to the posterior aspect. So this is the central grey matter. Okay, now this particular part is the central grey matter. Can you see the central canal? Okay, ये देखो, this is the central canal, around that this is the central grey matter. Okay, posterior to the central grey matter, okay, you can see some important nuclei are present. Now what are those nuclei? If you go from medial to lateral, what are those nuclei? So here you can see first is the nucleus gracilis, you can see the nucleus cuneatus, and if you come more laterally then you can see the spinal tract and nucleus of the trigeminal nerve. Spinal nucleus and tract of the trigeminal nerve. So like this, from medial to lateral if you go, you can see very clearly defined the nuclei. Okay, so here you have to mention about the connection, because here you can see the nuclei very clearly, that's why you have to mention the connections also. Okay, so nucleus gracilis and nucleus cuneatus become more pronounced. Okay, or you can say they are separated from the central grey matter and clearly the nuclei can be seen. Lateral to the cuneate nucleus, you can see the accessory cuneate nucleus also. See, this is the cuneate nucleus. Not lateral, the posterolateral part, you can see the accessory cuneate nucleus also. Okay. Now, which is equivalent to the Clark's column. Next, the spinal tract and nucleus of the trigeminal nerve can also be seen, which is more lateral to the nucleus cuneatus. Okay, that can be seen. So see, by looking at the diagram, you can write the position of the nuclei. Okay. Next, in this particular level, you can see the lower part of the inferior olivary nucleus. The entire olivary nuclear complex cannot be seen, but some part of the inferior olivary nucleus can be seen over here. Okay, crumpled bag appearance. Though olivary nucleus होता है, main olivary nucleus, the appearance is crumpled bag appearance. Okay, and it is having a hilum which is faced medially. So this is the hilum of the inferior olivary nucleus which is faced medially. Okay. Next, so these are the nuclei which you can see in the diagram. Okay, now these nuclei, what happened, they have been separated from the central gray matter. Okay, and they are placed at various position, okay, which we have seen from medial to lateral. Now if you concentrate on the central gray matter, this is the central gray matter. Now in central gray matter also you can see different nuclei are present. Okay, for example, nucleus of the hypoglossal nerve, dorsal nucleus of vagus, and nucleus of tractus solitarius. Okay, these are the three groups of nuclei which are present in the central gray matter, that means around the central canal. ठीक है, तो ये देखो, this is the central canal. Around the central canal these three nuclei are seen. Okay, the three sets means in right side also three, left side also three. Okay, or you can say three pairs of nuclei are seen. Okay, so these three pairs of nuclei this will be from anterior to the posterior, because if you come towards this side, towards the pyramid, that will be the anterior, and if you go away from the pyramid, then that will be the posterior. So from anterior to posterior in the central gray matter you can see these three nuclei. Okay, so that is all about the gray matter. Next, what we have to discuss after gray matter? Once gray matter is done, then what is the next? See, I have written gray matter के बाद क्या आता है? White matter. In white matter, what you have to know? You have to know about the fibers. Okay, what are the different fibers you see over here? तो यहाँ सबसे ज़्यादा important feature कौन सा है? Which is the most important feature? What is this label at the sensory decussation? तो यहाँ पे सबसे important feature कौन सा होगा? Which will be the most important feature? Sensory decussation. Okay, the fibers which are decussating at the midline, that is the sensory decussation. That is the most important feature, or you can say most important striking feature at this level. That's why we are studying this particular level. Okay, now before going to this, first of all you have to know that there are some fibers, they got specific names. For example, internal arcuate fibers. Okay, now see, these are the internal arcuate fibers. Now this arises from gracile and cuneate nuclei. Okay, gracile and cuneate nuclei, from there the fibers which are arising, these are called as internal arcuate fibers. Okay, now have you studied the pathway of pain pathway? You have studied the pain pathway. Okay, and you have also studied the different types of sensation carried. ठीक है, तो this gracile and cuneate nuclei, they are present, they are at which number neuron? First order, second order, third order? If you have studied the pathway, you must know. Okay, so what happens, the fibers which are coming out from the gracile and cuneate nuclei, these are called as internal arcuate fibers, as you can see, which are denoted as red in color. जो रेड कलर में दिखाई दे रहा है fibers, these are internal arcuate fibers. Okay, now this conduct the sensation of discriminative touch, position, vibration, etc. Okay, so these are the sensation carried by these particular fibers. Now they codes forward and medially around the central gray matter. You can see they codes forward and medially around the central gray matter. After that, what they will do? They will decussate in the midline. Okay, so these are the internal arcuate fibers. They are coming forwards, downwards, and medially around the central gray matter. Just below the central gray matter, they will decussate with each other in the midline. After decussation, they will ascend. Okay, now when they are ascending, they form a structure, and that is called as medial lemniscus. Okay, if it is one, that is called medial lemniscus. If it is two, then it is called as medial lemnisci. ठीक है, so ये देखो, this is the medial lemniscus. Next, can anyone tell me, ये जो medial lemniscus form हुआ है, when it is ascending, where it will go? It will go to the thalamus, as simple. And from thalamus, where it will go? It will go to the sensory cortex of the brain. okay, where the perception of the touch will be felt. Okay. Next, in this deposition, the gracile fibers are medial and cuneate fibers are lateral. जो deposition होता है, gracile fibers are medial, cuneate fibers are lateral. Next, in the lemniscus, the body is represented. Now, this is very important. ये जो lemniscus form हुआ है, the medial lemniscus, in that the fibers which are present, the lemnisci are formed by the fibers only. Okay. Now here the body is represented. Now how the body is represented? Head is posteriorly and feet is anteriorly. Okay. So a bundle of fibers are there. Anterior fibers are also there. Posterior fibers are also there. Okay. So the head which is there, head part का जो fibers है, that is present posteriorly, and feet part का जो fibers है, that is present anteriorly in the lemniscus. This is the representation. So what is the significance? If any damage is there in the lemniscus, okay, which part is damaged? If anterior part is damaged or posterior part is damaged, based on that the sensation loss will take place, because these are the sensory fibers. Okay. Next, the accessory cuneate nucleus is also there. You can see the accessory cuneate nucleus, which is dorsolateral, or you can say posterior to the nucleus cuneatus. Okay. So what is this accessory cuneate nucleus? The accessory cuneate nucleus receives fibers from the cervical segment of the cord, okay, of the fasciculus cuneatus, and they give rise to posterior external arcuate fibers. Okay. So ये जो accessory cuneate nucleus hai, from here posterior external arcuate fibers are coming out. Okay. Now they convey the proprioceptive impulses to the cerebellum. That means from the accessory cuneate nucleus, the fibers which are coming out, they are carrying the proprioceptive sensation. Okay. And this proprioceptive sensation will go to the cerebellum. How they will go to the cerebellum? By inferior cerebellar peduncle. In cerebellum, I hope you have studied about the peduncles. 3 peduncles are there in the cerebellum. Okay. तो क्या होता है, जो brainstem है, 3 parts of the brainstem are connected to the cerebellum by 3 peduncles, superior, middle and inferior peduncles. So medulla connected to the brainstem, cerebellum by inferior cerebellar peduncle. Okay. So these fibers, posterior external arcuate fibers, they are carrying the proprioceptive sensation and they are reaching the cerebellum. How? By inferior cerebellar peduncle. Okay. Next, the pyramidal tracts. You can see the pyramidal tracts are lying anteriorly. Okay, over here. Then if you see posterior to the decussation, okay, so this is the decussation, just posterior to the decussation. Can you see these 2 structure? This is called as medial longitudinal bundle. Medial longitudinal bundle, which is situated posterior to the medial lemniscus. Now, what is the function of this medial longitudinal bundle? Now, it is a small compact group of fibers which interconnect the 3rd, 4th, 6th, 8th and 11th cranial nerve nuclei. Okay. So these cranial nerve nuclei are connected to each other by this medial longitudinal bundle, and it is having very important function. Now what are those functions? It integrates the information received about the movement of eyes and movement of head. Okay, whenever the head is moving and whenever the eyes are moving, at that time it plays a very important role in controlling the optokinetic and vestibulo-ocular reflexes. Okay, I will give an example of this medial longitudinal bundle. Suppose whenever you are doing any activity where head movement is there, okay, if you are doing any activity where head movement is there, at that time also you can see the object. I will give an example. Whenever you are playing, suppose you are playing cricket, whenever you are playing cricket, suppose ball is coming towards you, you have to run and you have to catch the ball. ठीक है, at that time head movement भी हो रहा है, you are looking at the ball continuously. Okay, head movement हो रहा है, but the vision is clear, you can see the ball and you are going to catch the ball. So that coordination which is taking place, okay, that is called as optokinetic and vestibulo-ocular reflexes. and in that the medial longitudinal bundle is helping. Okay, so it is helping to see any object which is in the field of vision but it is moving. Okay, and it is maintaining the head movement as well as the eye movement, so that the object which are in movement, okay, can be seen easily. So that's why I am given an example. Whenever you are playing cricket, you are trying to catch a ball, you are running, head movement is also taking place, eye movements are also taking place. Okay, so that particular coordination is done by the group of fibers and it is called as medial longitudinal bundle. Okay, medial longitudinal bundle or MLB. This is the medial longitudinal bundle, this one. Okay, next, so in the white matter, after medial longitudinal bundle, next is the spinocerebellar and lateral spinothalamic tracts are present. Okay, and anterior and lateral spinothalamic tracts. So see, these are the spinothalamic tract. Here you can see the spinocerebellar tracts also. So जो anterior and lateral spinothalamic tracts है, you have studied about the tracts, right? Ascending tracts, descending tracts, it's done. I know you have studied. तो जो anterior and lateral spinothalamic tract है, can you tell me the difference between the anterior and lateral spinothalamic tract? Anyone? Anterior and lateral spinothalamic tract difference क्या है? Yes, yes, yes. Yes, very good. Now, do you know one thing? Anterior and lateral spinothalamic tract together, okay, it is called as spinal lemniscus. Anterior and lateral spinothalamic tract together, they are called as spinal lemniscus. Okay, so whatever you have studied, please add this line that together they are forming the spinal lemniscus. Okay, so that is the white matter which are present over here. Now see, in white matter, what we have discussed, first of all, the most important one we have seen, that is the internal arcuate fibers over here. Okay, the deposition of the internal arcuate fibers which are forming the, what is this? This is the medial lemniscus. Okay, that is the sensory information, different sensation. Next, we have seen the accessory cuneate nucleus. Which sensation are carried by the accessory cuneate nucleus? Proprioceptive sensation. Okay, and from accessory cuneate nucleus, this sensation are going to the cerebellum by inferior cerebellar peduncle. Okay, next, which fibers we have seen? We have seen that, what is this? Medial longitudinal bundle. Okay, the importance of the medial longitudinal bundle I have mentioned. Next, you can see the lateral spinothalamic tract. So lateral and anterior spinothalamic tract will form the spinal lemniscus. Okay, and you can see the other tracts, spinocerebellar tracts on both the side. Okay, so these are some important fibers. Okay, white matter which can be seen in this particular section. So this section is at the level of sensory deposition. Okay. Next, the deposition is at the level of olive. Okay, next deposition is at the level of olive. So in the diagram I have shown you the inferior olivary nucleus, if you have seen. So complete olivary nuclear complex is not seen at this level. For that, what we have to do, we have to take a section at the level of olive. Okay, so here you can see, if the transverse section is taken at the level of olive, it passes across the floor of fourth ventricle through and the middle of the olive. Okay, so this section will pass at the level of floor of fourth ventricle. Okay, and along with that, it will pass to the olive also. So though it is passing to the floor of fourth ventricle, at that time, what we can expect? We can expect to see the structures of floor of fourth ventricle also. Okay, and along with that, we will see the olive. Now here you can see the diagram. So with the help of this diagram, we will try to understand the structure. See, as we are going upward, okay, the structures, or you can say the TS will become more complicated as we are going further. The structures will get more complicated. Okay. Now here also we have to segregate. We have to see the gray matter. So here you can. This is the floor of fourth ventricle. Okay. This is the floor of fourth ventricle. So in the beginning, what we have to do, we have to identify which is anterior side and which is posterior side. Okay. So see, pyramids are still seen over here. So that is very easy for us to identify which is anterior and which is posterior. तो देखो यहाँ पे पिरामिड दिखाई दे रहा है, मतलब ये जो साइड है, दिस विल बी द एंटीरियर साइड, ओके, एंड दिस विल बी द पोस्टीरियर साइड. Now here this particular area is forming the floor of fourth ventricle, okay, because we have taken the section through the floor of fourth ventricle and through the olive. Okay, so here you can see the entire olivary nuclear complex. ये देखो, entire olivary nuclear complex can be seen very easily. Okay, so we have identified which side is anterior, which side is posterior. Okay, now what is our job? Now our job is to identify the important nuclei which are seen at this level. Okay, so this black color dots, as you can see, these are nothing but the important nuclei. All the nuclei are seen over here. So here also same, the nuclei we have to identify from medial to lateral. Okay, so central gray matter is spread. Okay, the central gray matter, you can see it is spread over the floor of fourth ventricle. Okay, and it contains different nuclei of cranial nerves. From medial to lateral, these are hypoglossal nucleus. You can see the hypoglossal nucleus. If you go lateral to hypoglossal nucleus, you can see nucleus intercalatus. See this is the nucleus intercalatus. Okay, then you can see the dorsal nucleus of the vagus. Then you can see the nucleus of solitarius, tractus solitarius, and here you can see the vestibular group of nuclei. So like this, from medial to lateral, okay, all the nuclei you can see at a row. So mostly they are situated lateral to each other, but the position is going towards the posterior aspect also. Okay. Now can anyone tell me what is the function of this nucleus of tractus solitarius? Nucleus of tractus solitarius. For that you should know about the taste pathway. Okay, for that you should know about the taste pathway. तो जितना भी taste sensation है, okay, from the tongue, various part of the tongue, from the epiglottis, from the soft palate, all are carried by this nucleus of tractus solitarius. So this is the important nucleus of the taste pathway. Okay. Now, so here you can see all the nucleus. Now this is the pontomedullary junction. So close to the pontomedullary junction, dorsal and ventral cochlear nuclei can be seen. Okay, you can see the cochlear nuclei also. See, the cochlear nuclei can also be seen. So these are the different nuclei can be seen. Next, nucleus ambiguous is also seen. See, this is the position for the nucleus ambiguous, and if you see the position of nucleus ambiguous, it is situated deep in the reticular formation. Okay. Now this nucleus ambiguous gives motor fibers to 9th, 10th and 11th cranial nerve. Okay, 9th, 10th and 11th cranial nerve का जो motor fibers है, those fibers are coming out from the nucleus ambiguous. Okay, and along with that, here also you can see the spinal nucleus and tract of the trigeminal nerve that can also be seen. Okay. Now, can anyone tell me which is the most striking feature of this particular area? Every time, whenever I am discussing about any of the section, I am mentioning the most important striking feature. So, in this section, which is the most striking feature? Yes, olivary nuclear complex. This one, ये पूरा, this one. This is the most important feature of this particular area. ठीक है? So what you have to know about this inferior olivary nuclear complex? So inferior olivary nucleus is the largest mass of gray matter seen in this level. Okay, and because of this there is a elevation in the medulla oblongata which can be seen from external aspect. Okay, that means on the anterior aspect of the medulla you can see the olive. Why? Because of the inferior olivary nucleus which is present deep to the olive. So it is the largest mass of gray matter which is present at this particular level. Okay. Now if you see the appearance, so as I said, it is crumpled back appearance and it is having a hilum, and that hilum is facing medially. Okay. Now close to the inferior olivary nucleus you can see the medial and dorsal olivary accessory nucleus. Okay. So you can see the medial and dorsal olivary nucleus and medial accessory olivary nucleus. So these nuclei together they are forming the olivary nuclear complex. Okay, different types of nuclei are there. So this is the main olivary nucleus. See, this is the main olivary nucleus. Then this is the dorsal accessory olivary nucleus, and this is the medial accessory olivary nucleus. Okay, together they are forming the olivary nuclear complex. Now the question is, इतना बड़ा nuclear complex है, then इसका function क्या है? What will be the function? So it is having a significant function. Unnecessary, the nuclear complex will not be there. So it receives afferent fibers from cerebral cortex, red nucleus, and periaqueductal gray of the midbrain. So these are some areas from which it is receiving afferent fibers from cerebral cortex, red nucleus, and periaqueductal gray region of the midbrain. These are the areas from which the fibers are coming into the inferior olivary nuclear complex. Okay? After that, what happens? The fibers which are coming out, those will be the efferent fibers. Okay? Now these efferent fibers form the olivocerebellar fibers. The efferent fibers are forming the olivocerebellar fibers which will go to the cerebellum and they terminate as climbing fibers. Okay, the climbing fibers which we have studied in the cerebellum chapter. Okay, तो ये जो inferior olivary nuclear complex है, from here the fibers which are coming out, they will form the climbing fibers that will go into the cerebellar cortex. Okay, and you have seen it is synapsing with the dendrites of the Purkinje cells. Okay. Next you can see the arcuate nuclei. ये देखो यहाँ पे एक arcuate nucleus रहता है both the side. Okay, you can see the arcuate nucleus. Now they thought to be inferiorly displaced pontine nuclei. Okay, the arcuate nucleus lie anteromedial to the pyramidal tract. So pyramidal tract का भी anteromedially, okay, the nucleus which is present, it is called as arcuate nucleus. Okay. Next, in this particular section, you can see another structure that is called pontobulbar body. ये देखो, okay. The elevation is there. It is called as pontobulbar body. Now it is located to the dorsolateral surface of the inferior peduncle. So this is the inferior cerebellar peduncle. Okay, dorsolateral, that means posteriorly and lateral to the inferior cerebellar peduncle, the pontobulbar body is present. Okay, pontobulbar body. Now what is this pontobulbar body? The pontobulbar body receives afferent from corticopontine fibers, circumolivary bundle, and gives efferent to the cerebellum via inferior cerebellar peduncle. Okay, so what it will do, it will receive fibers from corticopontine fibers, that means from cerebral cortex, then pontine nuclei, and then fibers are going into the pontobulbar body, and from pontobulbar body the efferent fibers which are coming out, they are going into the cerebellum, okay, via inferior cerebellar peduncle. So that is the gray matter. Next is the white matter. Now for white matter, this time what I have done, I have added one nice diagram. Yeah. So here you can see this is also at the level of olive, but here you can see the fibers, especially about the fibers. That means particularly from which nuclei, which fibers are coming out, that you can see in this particular diagram. Okay. So inferior cerebellar peduncle is occupied posterior lateral part of the fourth ventricle. So this is the fourth ventricle. So posterior lateral part of the fourth ventricle, here you can see inferior cerebellar peduncles are present. Okay. Now olivocerebellar fibers are seen. Okay. So olivocerebellar fibers, see, these are the olivocerebellar fibers, okay, which are going as climbing fibers. Then stria medullaris are also called as anterior external arcuate fibers. Okay. So here you can see the arcuate nucleus. From here the fibers which are coming, these are called as stria medullaris of the fourth ventricle, or also called as external anterior external arcuate fibers, okay, which are present in the floor of the fourth ventricle. Now this arcuate nucleus receives fibers from the cerebral cortex and send efferent fibers to the cerebellum. Okay. Opposite to the anterior external arcuate fibers. Here you can see anterior external arcuate fibers which are going to the cerebellum. Then emerging fibers of 9th, 10th and 11th cranial nerves are also seen. You can see fibers of the 10th cranial nerve can be seen over here. Okay. So like this 9th, 10th and 11th cranial nerve, the emerging fibers can be seen in this particular view. Okay. So that is all about the internal structure of medulla. Okay. So total 4 sections are there. Among them we have discussed about the 3 sections only. One section is left, please study from your own. That section is not that difficult, it's very simple. Okay, that's why I have not included. Okay. Now, as you have noticed, once we are going upwards, the sections are getting difficult. Okay. So please keep that in mind. If you know the diagram, first of all you have to know the diagram and in which particular location which nuclei are situated. Okay, that you have to know. And the last most important thing is, if any important nuclei is present in that particular area, just try to know the connection. Okay. The best example which I have given is for the inferior olivary nuclear complex. Okay. Similarly, you have to proceed. Okay. Next is the pons. Okay. Internal structure of the pons. Now, before going to the internal structure of the pons, you have to know one thing. Okay. What is that? In case of medulla, identifying which is anterior side, which is posterior side, it was very easy, right? But same thing in case of pons, it is not that simple. For that, what you have to know? For that, you have to know about the internal structure of the pons. Now, when you are taking the section of the pons, okay, at that time, anterior side is there, posterior side is also there. But two different names are given. That is the basilar part, large ventral part called as basilar part. Small dorsal part is called as the tegmental part. Okay, two different parts are there of the pons. So, जो anterior part होता है, that is called as basilar part. Posterior part होता है, that is called as tegmental part. Now, please listen carefully. ये जो basilar part होता है of the pons, okay, the structure is same at any level. Generally, in pons we are taking section at 2 level. Upper part of the pons and lower part of the pons. Okay. So any section, if you see the structure, it is same for the basilar part, but for the tegmental part it is different. Okay. For that we have to take 2 different sections and we have to study it separately. Okay. Now in the ventral and the basilar part, continuous inferiorly with the pyramid. Okay. And dorsal tegmental part is direct upward continuation of the medulla, excluding the pyramid. Okay. So here you can see the basilar part is basically same at all the levels, but the tegmental part vary considerably, upper part and lower part. Now let me show you the diagram. Here you can see the diagram. Can you identify which is the ventral part and which is the dorsal part? Yes, so this is the ventral part and this is the dorsal part. So this is also called as the large basilar part. The ventral part hota hai, it is larger. Okay, dorsal part is smaller. Now the question is, what are the differences? Now if you see the basilar part, the anterior part, it is made up of longitudinal bundle fibers, transverse fibers and pontine nuclei. Longitudinal fibers, transverse fibers and pontine nuclei. These are the three structures present in the anterior aspect, okay, in the basilar part. Now what about the tegmental part? Now in tegmental part, mainly you will see the deposition of transversely running fibers, and those are called as trapezoid body. Okay, जो tegmental part है, I am talking about this particular part. Another diagram is there, wait I will show you. Here you can see the trapezoid body. तो जो anterior part है उसमें क्या रहेगा, the fibers, longitudinally running fibers, transversely running fibers and pontine nuclei, those are situated over here in the anterior part, that is the basilar part, large, ठीक है, and इसका structure same है in the upper part of the pons, in the lower part of the pons. If you go posteriorly, then in the upper part and lower part the structures are different, ठीक है, but what you can expect to see in the posterior part. So first, the most important thing which you can see is the transversely running fibers which are depositing, and those are called as trapezoid body. Okay, center mein rahega. This is the trapezoid body. Along with that, you can see the nuclei of 5th, 6th and 5th, 6th, 7th and 8th cranial nerve. Nuclei of 5th, 6th, 7th and 8th cranial nerve. Can you tell me the name of the nerves? 5th, 6th, 7th and 8th cranial nerve. Okay, so the nuclei of those cranial nerves are situated over here. Okay, so this is the difference between the anterior part and the posterior part. So I hope it is clear about the basilar part and the tegmental part of the pons. Basilar part is larger, tegmental part is smaller. जो cranial nerve nuclei है, those are present in the tegmental part only, posteriorly रहता है, okay? और जो basilar part है, उसमें क्या रहता है? Bundles of longitudinal fibers, transverse fibers and pontine nuclei, okay? And in tegmental part, another important structure, that is the trapezoid body which is present. So like this we can differentiate the anterior aspect and the posterior aspect. Another important point, if you have noticed, on the tegmental part, posterior part, you can see the small portion of the cavity of fourth ventricle. Can you see? ये देखो. Okay. Next, what we have to do? We have to study the sections of the pons. Okay. At the level, two different levels are there. Okay. Lower part and upper part. So this is the transverse section of the pons through the lower part, or also called as the section through facial colliculi. Okay, so this is the section of the pons which is passing through facial colliculi. You can see this is the facial colliculi. Facial colliculus is a very important structure seen at the floor of the fourth ventricle. Okay, so I will stop over here.